All right, let's talk about CRISPR. You've probably heard the name, but today we're going to get past the hype and really dig into how this incredible technology actually works. Let's get right to it. So everyone calls it genetic scissors, right? And that's a great metaphor, but it's also kind of simple. I mean, how do these scissors find the one exact spot to cut in a DNA code that's billions of letters long? And what if you don't want to just cut something out? What if you want to rewrite it? That's what we're going to unpack. Okay, first things first, we have to start with the basics, the classic system that really kicked this whole revolution off, CRISPR-Cas9. You know, this whole thing didn't just appear out of nowhere. The story actually starts way back in 1987. Scientists found these weird, repeating patterns in the DNA of bacteria, but they had no idea what they were for. It was a total mystery. It wasn't until 2012 that the real eureka moment happened. That's when researchers realized they could hijack this system and program it to edit genomes. I mean, think about that leap. It was so huge, it won a Nobel Prize just 8 years later. That's lightning fast in the world of science. So what is CRISPR-Cas9? Well, believe it or not, it's actually an ancient defense system. It's how bacteria fight off viruses, kind of like their own immune system. They basically grab little snippets of DNA from invading viruses, like a mugshot, so they can recognize and chop up that virus if it ever shows up again. And scientists, in a moment of sheer genius, figured out how to take that natural system and repurpose it for editing, well, pretty much any DNA. All right, so the classic system really boils down to two key parts. First, you've got the guide RNA. Think of this as a super precise molecular GPS. It's a little piece of RNA that you design in a lab to perfectly match the DNA sequence you want to target. It's the brains of the operation, and its whole job is to lead the second part, the Cas9 protein, right to that spot. Now, Cas9, that's the scissors. It's a special kind of protein called a nuclease, which is just a fancy word for something that cuts DNA. Once the guide RNA gets it to the right address, Cas9 goes to work and makes a clean slice right through both strands of the DNA. But wait a second, there's a catch, a really important detail that makes this whole thing work. See, Cas9 can't just latch on anywhere it wants. It needs, like, a secret handshake to even get started. And that secret handshake is called a PAM sequence, protospacer adjacent motif. It's basically a short, specific string of DNA letters. You can think of it like a password or a keyhole. The Cas9 protein is constantly scanning the DNA, but it won't bind, it won't do anything until it finds this exact PAM sequence sitting right next to the target. No PAM, no cut. It's a critical safety feature, but also a major limitation. And this is where things get really interesting, because this limitation was a huge deal. The most commonly used Cas9 protein, the one from Streptococcus pyogenes, it only recognizes one specific password, NGG. So what does that mean? It means you can't just edit anywhere in the genome. You're restricted. You have to find a place you want to edit that also happens to have that NGG sequence right next to it. Imagine there's a typo in the genetic code causing a disease. If it doesn't have that NGG password nearby, the original CRISPR-Cas9 just can't touch it. This one little requirement left huge chunks of the genome completely off limits, and overcoming that problem is what really sparked the next wave of CRISPR innovation. Right, so scientists had this amazing tool that was also kind of handcuffed. They needed more than just scissors. They needed to build a whole evolving toolkit, and that is exactly what they did. Okay, so remember, the original Cas9 just makes a clean cut, a double-strand break, and then it just leaves. It relies on the cell itself to stitch things back together. The problem is, the cell's repair crew can be a little clumsy. They often make mistakes, little insertions or deletions. Now, that's actually perfect if you want to just shut a gene off completely, what scientists call knocking it out. But what if your goal is more delicate? What if you need to make a precise single-letter correction? And this is where the toolkit expands. Enter base editing. This is a total game changer. Instead of breaking the DNA in two, base editors use a dead Cas9. Its scissors have been disabled, and they bolt on a whole new enzyme. This new tool doesn't cut. It performs molecular surgery. It chemically rewrites a single DNA letter into another, like turning a C into a T, without that dangerous double-strand break. It's all about precision. So how does this magic trick actually work? Well, first, that disabled Cas9 still acts as the GPS, taking the whole package to the right spot. Then this new enzyme, a deaminase, works its chemistry magic. It converts one base, say a cytosine, into something called uracil. Now the cell's own repair machinery sees this uracil, which shouldn't be in DNA, and thinks, whoa, that's wrong, and fixes it. But in the process, it finalizes the edit we wanted all along. And to make sure the cell doesn't just change it back, there's another little piece, an inhibitor, that locks the change in place. It's just incredibly clever engineering. But the evolution didn't stop there. Next came prime editing. If base editing is like a pencil with an eraser, prime editing is like the search and replace function in a word processor. It's even more sophisticated. It uses a Cas9 that only nicks one strand of the DNA, and it's fused to a different enzyme called a reverse transcriptase. This allows it to directly write new genetic information into the DNA at the target site. We're talking all 12 possible single-letter changes, plus small insertions and deletions. The versatility is just mind-boggling. Okay, so we have all this incredible power to cut, rewrite, and replace, but with that power comes a massive, massive challenge. You have to be absolutely sure you're editing only the right spot. And that brings us to maybe the single biggest safety hurdle in the entire field. We're talking about off-target effects. See, the genome is a massive place, and sometimes a sequence of DNA in a totally different location looks really, really similar to your target. The danger is that your CRISPR system gets confused, goes to one of these lookalike sites, and makes a cut or an edit where it's not supposed to be. And the consequences of that could be disastrous. You could accidentally turn off a critical gene, or even, in a worst-case scenario, trigger something like cancer. So how do you prevent that? Well, scientists don't just guess. They use really sophisticated computer programs to predict the risk. These algorithms scan the entire genome and generate a score, like this aggregated off-target score you see here. It's basically a number that tells you how clean your guide RNA is likely to be. And it's pretty simple to understand. A higher score is better, much better. A score up in the high 90s, like a 98 or 99, tells a researcher that their guide is probably very precise, with a very low risk of hitting any unintended targets. But if you get a low score, that's a huge red flag. You'd probably throw that guide RNA out and design a new one. But they didn't just stop at prediction. Scientists went back to the drawing board and actually re-engineered the Cas9 protein itself to make it safer. They've created these high-fidelity versions, which are way more accurate. For example, there's one called ESP-Cas9 that's designed to be less sticky with the DNA in general, so it's less likely to bind in the wrong place. There's another called SuperFi-Cas9 that's extra good at double-checking for mismatches before it cuts. And then there's Hypo-Cas9, which actually slows the whole process down, giving it more time to proofread its work before committing to an edit. It's a constant race for more and more precision. So all this incredible work on precision, on safety, on expanding the toolkit, it's all pushing CRISPR out of the research lab and into the real world. We're starting to move from just editing code to actually creating cures. And the level of control scientists have now is just staggering. It's almost science fiction. We can now build CRISPR systems that just float around inside a cell, totally dormant, until we switch them on with a specific trigger. That trigger could be a particular chemical we add, or, get this, even a targeted pulse of blue light. Imagine that. It gives us the power to control when and where a gene edit happens. The precision is just on a whole new level. So let's just recap. We've gone from a mysterious quirk in bacterial DNA to a programmable toolkit that can search, replace, and rewrite the code of life. A toolkit we can now control with a literal flash of light. The pace of this technology is absolutely breathtaking, and it leaves us with the biggest, most profound question of all. Now that we have this power, what do we do with it? With the ability to rewrite the very essence of life, what should we edit next?